Thank you to the organizers. I will say something about myself during my first slide. Felice this morning mentioned the fact that we are living in a transition period, and now here I'm showing how different aspects of medicine live together in this very moment. And they will continue to do so for a long time. Because the idea of using precision medicine for all diseases and all patients is something which is far to come and that we will have to assess more carefully. During my short presentation, I'll try to highlight some of these aspects. But as the chairman said, I will have to introduce myself. And I can say that before entering into the pharmaceutical world, I was a doctor for a certain period of time. In a period when these different layers did not exist, many only focused on this first part, and a physician was the person who would administer a substance he does not know, he knows nothing about, to a body he knows even less. A lot of time has gone by, and we have been learning many new things, but some problems are still standing, some of which are trivial problems, but you should be aware of these problems, because the first take-home message is the following one. Don't feel discouraged if in the passage from a scientific idea down to the concrete results you encounter obstacles, because resilience is something you have to keep for yourselves and use anytime or whenever it is necessary. Another aspect. It has been said here today that statistics or modeling may help us in getting to a good result. I would like to add a third element to these two initial elements, which is serendipity. In many cases, we do things without exactly knowing what we are doing, but the end result is positive. So in this case, too, my encouragement is you may get to very good results even when you do not know everything. The first aspect. I've been given this slide from my children because when I said I wanted to go back to the past to explain the future, the Back to the Future car came to their minds immediately. And the whole story started in 1973 when studying the chronic myeloid leukemia. A specific study detected the fact that in this specific disease, there was a role for translocation of chromosomes. At first, the chromosome 9 was at stake. Then it became apparent that a new oncogene was formed, which produced a protein activating the cells, a protein which gives a cell signal saying, do multiply and do not die. From a clinical point of view, this gives rise in terms to the cells of the hemopoietic system, So once in a tumor, a point alteration was identified when it was called Philadelphia chromosome, because the first translocation was identified in Philadelphia. Once that achieved, the researchers started to say, well, we have found the target and leukemia will soon be cured. But if you listen to what I've said, you can realize that this first discovery back in 1973, in that discovery, the first molecules which interfered with this kinase were identified back in 1992, many years later. So my message is that going from a scientific idea to the practical implementation takes time, and it can be very complicated and can be stopped by trivial problems. The Novartis, the company I'm working for, at a certain point identified some molecules which in vitro had an effect in inhibiting the tyrosine kinase. But unfortunately, molecules have to be soluble. In other words, you have the molecule, you are very happy, you think you have found this solution, but this molecule is not soluble, cannot be administered, in other words, or the molecule is not stable. As soon as you administer it, it changes and it's of no use anymore. The first thing that comes to our minds is that in many cases, simple things, trivial things, especially if compared to what we discussed this morning, may come in between the scientific idea and the implementation of the results. I have to list yet some more difficulties. So you find a stable substance you can administer to dogs in that case, and that substance caused thrombosis. And the experiment had to be stopped because the substance could not be administered, and the whole development process had to be reconsidered. But now we are in 1996. If you follow the whole development, and if you consider that we are now discussing chromatin and precision medicine, you consider here we are just trying to make a molecule which can be administered. So the test was started in 1998 on 12 patients. When the drug was administered to men, the dosage was very low because the first problem is not to cause havoc. So you start with low doses, the administration at low doses without knowing whether there is therapeutical effectiveness. So we had the first 12 patients, and the dose was little by little increased. And then we had the first clinical result. In some cases, science has to come to terms with trivial things. We have the first clinical effects at 140 grams, but the patient came to us to say, well, I cannot stay in the USA anymore. That was a clinical experiment in the USA because I want to go back to my family. I withdrew from the experiment because I want to go back from the family. All scientists in the world were considering this experience, and you have to come to terms with a personal problem. A person who tells you everything is fine for me, but I want to go back to my country. Now we are in 1999, and we have a strong result. A data telling us that the molecule can reduce the cells in the circulating system, but can also reduce the Philadelphia positive cells when counted in the bone marrow. That was the technology. I will tell you something about this later, because that was the system according to which the disease progression was measured. So we had in the first clinical data back in 1999. At that point, a new problem emerged, the problem of an industrial nature. You have a limited number of patients, but you know that it is very likely that the drug you are studying will change the cure of that and the care of that disease. But at first, the drug has to be produced by pilot plant, producing just a few kilos of the molecule. If you have to move on to produce the drug for clinical use, then you have to produce different, many hundred kilograms of the molecule. But you just have a few data on the patients, and you have to make up your mind. Are we going to build a plant on just 12 patients or not? In some cases, many believe I'm representing the big pharma world. And some could say, but big pharma is so rich that they can decide to open a plant whenever they want. But when you manage a company, if you invest in a sector, that means that you're not investing in another sector. You have to make a choice. And we chose to start with a plant without knowing whether the drug could be marketed in the end. And that choice was really exciting, an exciting choice, because even those who worked at that plant started to become passionate. They grew passionate and they started to work around the clock to try and produce the molecule in the fastest fastest way possible. Sorry.
Arriva sul mercato.
So everything is fine now. The drug is marketed, gets to the market. It's approved by FDA and by EMA in Europe. But as many of you may know, in Europe, one thing is the approval by EMA, which says that the drug does work, is scientifically proven and can be marketed in all the countries in Europe. That's one thing. And another thing is that each and every country may decide whether the national system pays for the drug or not. And in the UK, NICE has the last word. And NICE said, no, we want to have survival data before paying for the drugs. And this is a serious problem for precision medicine, because it's very likely that precision medicine is a kind of medicine which will enable us to get to more telling results, more suitable for the aim. But here the authorities said, in order to be able to approve the molecule, I have to have data about the survival data. At that time, the survival rate for chronic myeloid leukemia was 5 years. So in order to have data on survival, only if you were very lucky, you could have a result after 3 years. This is a letter written by a well-known hematologist who criticized that decision, and he said the drug is a great drug, you must approve it. NICE in the end said or decided to approve the drug to reimburse it, but 19 months later than all the others. And many people died of leukemia simply because the labs were not able to understand that when we move towards more sophisticated drugs and when we use biomarkers, something that tell us what is happening, then we do not need to wait for 5 years, and patients do not need to wait for 5 years, more in particular. But as I said earlier on, I'm not going to tackle problems only, because we also want to have serendipity. What do we learn, and what have we learned? In studying this disease and the molecule we used, we said about following this disease with cytogenetics is very imprecise. You harvest the material from the bone marrow, you observe it, you count the Philadelphia positive molecule, and from that you should understand whether there is progression in the disease or not. It's highly imprecise, highly painful for the patients. Not pleasant, really. So we started to wonder whether we could measure differently the presence of this altered protein with a different technique. Therefore, so that is why, and that is how we learned that that is possible, and that thanks to that molecule, the presence of this anomalous protein could be detected with a molecular system, even when it was present in very small quantities, because the method could become more sensitive. We also learned that there is correlation between the measurable quantity of this protein and the expectation of disease-free period for the patient. We learned a lot, but we also learned that if we could reduce the presence of this protein down to very low quantities, if we could reduce this co-gene presence at very low level, if we could reduce it, then maybe the patient could even withdraw the therapy, could be in remission for a longer period of time. This is the study of an experience published by a French group. In this group, those patients having a very low level of this protein were studied. The levels were kept low for more than 2 years, and then there was withdrawal, and then 40% of these patients did not incur in any recurrence of the disease. This is a fascinating aspect because this is telling us that within this balance, even the presence of some positive proteins, positives to this oncogene, could be kept under control by the system or by something which kept the upregulation under control. Professor Pellicci told us this morning that cell wants to exercise the freedom, and they do not want to follow something which is codified, but they want to be able to choose their direction. There must be Italian cells in most cases. But we have also learned that some of these patients have progression in all instances, and we have not understood why, really, because a patient who is in remission for 5, 7 years at a certain point has the progression of the disease and moves on to the plastic form with the disease which progresses and is no longer under control and becomes very aggressive. That is why we decided to look for other inhibitors of this anomalous protein. And some drugs were selected, drugs which seem to be interacting with the protein. They were started with the normal inhibition tests. And then we saw that the co-administration of the first molecule, imatinib, with a second molecule from the very beginning, worked in such a way that certain patients who had the disease out of control could see the disease getting under control again. This paves the way for new results and new considerations. But the most interesting aspect here is that this is telling us that it's very likely that some clones which have a mutation are there from the very beginning. And therefore, if we manage to identify with precision medicine which are the patients already having these clones, may enable us to treat these patients from the very early stage, from the very beginning, thus obtaining better results, or good results. This, of course, opens up a new stream of thinking. The idea of using more substances to try and interact with a greater number of pathways, as if you were in a street of a large city, a big city, and you are in a bottleneck because of a traffic jam, and you try to find a way out, another way. And cells are not so different from human beings. They have the same reactions. Even tumor cells behave like human beings, which because of negative stimuli reacts aggressively. And cells do the same. They find sideways. And this makes us think that we could block more than one pathway. We could create more traffic so that all cars are stopped, are blocked in the traffic jam. This is what we are doing. This is the approach to the melanoma therapy. We apply 2 blocks. We have some clinical evidences which have been recently published showing that the use of 2 substances is better and gives better results than the use of 1 substance alone. And we are developing models on the basis of the size of the tumor, of the multiplication rate of the tumor, and many other factors. So precision medicine in the oncological field may be of great help to overcome the drug resistance, for example, to enable us to understand how side effects can be reduced. So it's a highly promising revolution or new approach. But precision medicine also brings about with it organizational problems. You cannot think of a patient going to a hospital to be tested for a specific mutation which is present in 5% of the population. So the patient is negative, and then he goes to another hospital because the other hospital has another test for another mutations. That is why we have to create a network of centers that can be part of a screening, which is already the case in France. The national system to fight against tumors in France is organized in such a way that all centers are connected together, and they can perform an overall screening of the mutations. This allows for another solution. All drugs can be sent to that center because once the patient has been selected, the patient is followed in a certain center and gets the best therapy and the best treatment available. Can we really think that everything goes in this same direction? Well, this is one direction. But yet again, we have to overcome some trivial obstacles because this precision medicine can improve the results obtained. But when you discuss with the health systems, then you have to discuss on the basis of a yearly budgets. The long-term prognosis is not really taken into account, or the health systems do not consider the balance between the single cost and the reduction and containment of the overall costs. So we have to think and to change the way in which we think. We have to think differently. I've been very much impressed by an experience that has also been mentioned already. So I've been impressed by our work with Google and by the way in which they think. Let's consider the automobile industry. We know that from the economic point of view, and also in terms of mortality, we have a serious problem with car accidents. It's the highest mortality death cause for people under 40 years of age. So the car industry has improved the safety measures in the cars, airbags and so on and so forth. So Google approached the problem in a fascinating way, really. Google said, what is the cause at the basis of 90% of the accidents, of the car accidents? The driver. So if we do away with the driver, rather, if we replace the driver with something else, which is something which is not speaking on the cell phone, for example, then we may succeed in doing away with 90% of the car accidents. Google took a completely different approach from that of the experts from the car industry. They took a different approach. In precision medicine, too, we have to change our way of thinking. Instead of having the same approach we have from left to right, we have to go from right to left. We have to set the target, our aim. And then once the aim is set, then we have to reorganize the whole system in view of the aim set. Of course, this calls for organization, for cooperation. We have to do away with consolidated and sclerotized powers. But since I'm here with many young people, my invitation, please break away with the past. Do think from right to left. Lift all barriers to build the new medicine, because this is what can provide serendipity to all of us, and this is what should be done and ought to be done.